So firstly, I'd just like to thank the organizers for the opportunity to speak here today. As you can see, the topic I'm going to be discussing is the important role, emerging role of the microbiome in the context of psychiatry and neurogastroenterology. So just a quick disclosure slide before we move on to the main business of the day. And in his short story by Oscar Wilde, the protagonist needed to secure a red rose from the garden in order to fulfill their romantic ambitions. And alas, there were no red roses. So he cried out, on what little things does happiness depend? And I guess in the middle of a pandemic, I don't need to convince you that it is the little things indeed that make a very big difference to our mood and emotion. Now, of course, the microorganisms in our gastrointestinal tract are emerging as very important players as well in our happiness, and we know a lot about these microorganisms, particularly the bacterial component of it. And it's somewhat of a magnificent beast when you think about the numbers that are involved, with almost as many microbial cells as human cells, and maybe more importantly, a vastly increased number of genes compared to the human genome. And the real excitement, I suppose, is because this is a additional genome that we didn't know too much about up till recently, which is modifiable, and that might have important implications then for therapeutic targeting in various disease states. So although for a long time these microorganisms, this virtual organ, the microbiota, was neglected in a way, it's certainly not a neglected organ anymore. It's not forgotten anymore, and it's been in the spotlight in a number of different influential public domains. So why then would we think, as suggested in this article here in Science, that we might be able to mine the microbiome for drugs which could impact on brain function and behavior? And that's really what I'm going to talk about today, just to give you an introduction to the concept that the microorganisms which are resident in our gastrointestinal tract have a scope of influence beyond just a general impact on health and disease, and have an impact specifically on aspects of brain function, structure, and behavior. So I'm going to go through briefly, just to give you a quick whistle-stop tour, a snapshot of some of the important preclinical research and the signaling pathways that we think are important in the context of neurogastroenterology. And then in part two, I'll go more into efforts to see if this work will translate from bench to bedside, the efforts to see if we can get a more granular detail on the mechanisms involved, and to talk about then some of the opportunities that might arise from for intervention from these observations. So the reason the microbiota might be able to impact at higher brain centers is because of this framework provided by the brain-gut axis, or the gut-brain axis, whichever way you prefer. And really, this axis, knowledge about it has been around for quite a long time. It's just that relatively recently that the microbiota has been stitched into it. Now, because the brain-gut axis controls a variety of both basic and complex functions, the fact that the microbiota might be able to hijack the signaling pathways involved then gives a lot of scope for it to influence a vast array of host physiological aspects. And there are a number of key pillars in this gut-brain axis. So, for example, the gastrointestinal tract is very heavily innervated by the vagus nerve, and that is a direct route of communication between the gut and the brain. There are also important neuroimmune and neuroendocrinological components, like the hypothalamic pituitary adrenal axis, for example. And also, we need to realize that these microbes are like mini factories churning out microbials like short chain fatty acids, which can also directly impact host physiology. And really, a variety of different experimental approaches then to modify the gut microbiota have converged to give us the information whereby we think that the microbiota may be important for a range of behaviors, including those relevant to anxiety, depression, pain, and cognition, for example, as well as others. And many of these experimental approaches may be considered somewhat intuitive in that administration of probiotics or antibiotics, for example, but maybe one that deserves a little bit more explanation is the use of germ-free animals. So again, the germ-free animal is not a new model. It's something that's been around quite a while, but it's just relatively recently that it's been applied to the types of questions that we've been asking. And although you could argue that it's difficult to see a direct translational equivalent where there's such a complete obliteration of the microbiota across the lifespan, it still is a really neat model to get a yes/no answer on whether a particular aspect of brain function and behavior is altered as a consequence of growing up germ-free, and also whether that aberrant phenotype that you've noted could be rescued via different interventions. So, for example, reintroduction of a complex microbiota post-weaning, or monoassociation of the animals with particular strains that you might think to be relevant. And really, these germ-free animals have told us quite a lot about the scope of influence of the microbiota. So here we can see, for example, germ-free animals showing lower anxiety-like behaviors, and that tallies with what we know from probiotic studies, which indicate that the administration of probiotics can reduce anxiety-like behaviors. Again, indicating this is a behavioral domain that can be influenced by our gut microbiota. Now, we're all familiar with the stressors of modern living, and you can see both across the pond in the States, there's been quite some stressful scenarios lately. Of course, the fallout from Brexit is also occupying minds here as well. And in the middle of a pandemic, the stressful nature of the public health interventions that are necessary to control that outbreak are also a significant stressor for a large number of the population. Now, really what we're saying in the context of stress of those, it's not the stress per se that kills it, it's how we react to it. So we all encounter these different stressors on the roller coaster of life, but it's how we treat those and how we react to them as individuals that determines their impact on our health and well-being. And from a biological perspective, at least, how we react to stress exposure seems to be heavily dependent on the gut microbiota. So again, this is studies from germ-free animals showing that if you stress out a germ-free animal, that you get hyperactivation of the core stress response system, this hypothalamic pituitary adrenal axis, as indicated by corticosteroid production. And again, this tallies with what we know from other interventions that have been used in this regard, whereby probiotics or certain probiotic strains have been shown to reduce stress-induced corticosteroid production. And again, the scope and range of influence and the study of that, of the role of the microbiota, has extended as well to show that not just behavior and aspects of host physiology, but also actually right down to brain structure are influenced by the microbiota. So we can see here, for example, that there are specific nuclei within the amygdala, which is a very important brain region, excuse me, very important brain region for fear and anxiety. We can see there are specific nuclei that are enlarged or as well as hypertrophy of specific neurons, even as a consequence of growing up germ-free. Now, I could show you data for other important brain regions as well, like the hippocampus or the prefrontal cortex, which are very important brain regions in psychiatry. But suffice it to say that germ-free animals are markedly different as a consequence of missing their microbiota, and that it's possible to conceive how these different behavioral profiles that arise might be linked to specific abnormalities in brain regions and circuits that might underpin these different behavioral profiles. Now, in terms of one example of a mechanism that might be important in explaining these observations, my lab was very interested in the role of our gut microbes in regulating tryptophan metabolism. So tryptophan is an essential amino acid. It has to be supplied in the diet. You can see some of the foodstuffs that's available in there on the right. And really, it's a very important raw material for a number of bioactives and signaling molecules in the gut-brain axis. And that includes host pathways like serotonin, but also the microbes can utilize tryptophan to make metabolites, which can also have an impact on physiology. So tryptophan, as I've indicated, is a very important precursor to serotonin, which is maybe its best known role, because serotonin, of course, is the therapeutic target for most successfully deployed antidepressant therapies. But of course, there's a large reservoir of serotonin in the gastrointestinal tract as well, where it observes a lot of basic functions relating to motility and secretion. Now, again, studies in our germ-free animals showed that as one of the consequences of growing up germ-free is that you've got an increased supply of circulating tryptophan concentrations, and that also tallies then with what we see in the hippocampus of these animals, where there's elevated concentrations of serotonin and the main metabolite 5-hydroxy-indolacetic acid. Now, of course, tryptophan is a precursor for other bioactives as well, including those that the microbes can produce, like indoles and tryptamine. And there are other host pathways that are also of importance. For example, conversion of tryptophan into kynurenine, many of whose downstream metabolites also have neuroactive potential. And it's important to note as well that the serotonergic system and signaling within the gut-brain axis can be modified as a consequence of stress exposures. And that if you stress out a germ-free animal, you can see a very different response in the serotonergic system compared to an animal with an intact microbiota. And that manifests at different levels of the microbiota in different ways, and also in a somewhat sex-dependent manner. So all of this together then leads us to think that maybe, for example, we might be able to develop what we would call psychobiotics, which could be different microbiota-directed interventions to try and control how our microbes regulate tryptophan availability and metabolism, either in host pathways or also in microbial utilization of tryptophan. So I suppose where we're at in many ways in the field is that there's this very promising body of preclinical research which indicates the huge extent and scope of influence of the microbiota on brain function and behavior. And what we really need to try and get at now is whether what we're seeing in our preclinical studies is going to move well from bench to bedside. And there are always, of course, challenges for translation in psychiatry and neurogastroenterology. Some generic reasons why maybe not everything we observe in animals might translate effectively to humans. And also some specific challenges in the context of gut microbiota research. So just to summarize the first part of this talk, again, I've shown you that there are some lot of promising preclinical research, which will lead into the clinical research that I'm going to talk about in part 2. And what this research indicates is that the microbiota is very important in regulating behaviors and physiology relevant to both psychiatry and neurogastroenterology. So I've also highlighted some of the general signaling pathways as well as some of the specific examples that are important in the context of the microbiota. So in part 2 now, I'm going to go on to outline some efforts that have been made to see does this research effectively translate from bench to bedside. I'll outline some more of the mechanistic insights that have been obtained and attempts to see whether the compositional alterations that have been noted in these disorders might have a role in symptom generation, as well then as to think about some of the therapeutic opportunities that might arise from these observations. Let's just finish part 1 of this with just some acknowledgements of the larger Laboratory of Neurogastroenterology back here in APC Microbiome Ireland at University College Cork. And thank you for your attention.